Good evening, Dr. Chaudhary. Am I audible to you? So today we have a session on RNA-seq analysis, and we have with us Dr. Saurav Chaudhary, who is currently working as a postdoctoral research associate at School of Biosciences, Cardiff, University, United Kingdom. Prior to that, Dr. Saurav Chaudhary earned his doctoral degree from biotechnology from Indraprastha University, New Delhi. His experience includes principal research fellow at Canterbury Christ Church University, United Kingdom, and his area of interest include molecular biosciences, genomics, transcriptomics, and epigenomics. He is a specialist in next generation sequencing data analysis. He has into his credit numerous publications. So without wasting too much of time, I am handing over session to Dr. Chaudhary. Over to you, sir.
This meeting is being
You are
recorded.
not audible. I think you are mute, Dr. Saurav. You are mute, Dr. Saurav.
Hello, sorry about that. Now you can hear me.
Yeah.
Yeah. Okay. Thank you, Praveen. Thank you for the introduction. And yeah, so I'm going to share my slide first and then start. Is I'm the co-host, Praveen?
You are co-host. You can share your screen.
Okay. Can you see my screen now?
Yes.
It's a blank. Yeah. Okay.
Yeah.
Okay. Let me. And now you can see my presentation. Yeah.
Yes, Dr. Saurav.
Okay.
Visible.
Okay. Hello there, and welcome to another session of this Bioinformatics training course. And I hope you all are enjoying the training so far. And to continue, I will be with you for the next two sessions to understand the transcriptional dynamics using the RNA-seq data. And so what I'm going to do is in this the 2-day sessions that nowadays, everybody knows that there are a number of tools and number of scripts available online, and you can use those scripts and tools to analyze your data. But the main problems comes out to be how you are going to represent your data in your publications and how you are going to interpret that data. So my main focus on these two sessions are how we are going to interpret that data from the RNA-seq data after the analysis, and how we can generate the figures for our data. So in addition to introducing the tools which I'm going to use for the RNA-seq analysis, I will also tell you that about how to read your output files and how to mean actually in terms of biology about those output results. And if you have any query, just put in the chat box and we will take all the queries after this session. And you can also, if I don't get time to respond back, you can also email me, which is you can see. Let me open my pointer. It's here, and you can note it down. I will put in my chat box as well my email address, and you can contact me in my email. So let's moving on. Before I start going further, I would like to introduce myself. I'm Saurav Chaudhary, and I did my PhD from Guru Gobind Singh Indraprastha University, Delhi, and followed by various research positions. And currently now I am research associate at Cardiff University, And you can contact me, or you can get connected with me of any one of the research profile nowadays. You can use ResearchGate, Google Scholar, or KidID, and you can also email me if you have any questions, and if you are interested in what I'm doing in my research, you can also contact me on my email address. Okay, and so moving on, this session, who is going to get benefit from this training? And I think from my point of view, if you are um, doing research in molecular biology, so you have to be good in wet lab experiments, as well as the knowledge of some bioinformatics tools to analyze your data. So from this session, the researchers who are geneticists or molecular biologists who don't have any idea of bioinformatics will be beneficial for them. This session is going to be the beneficial for them, as well as for those bioinformaticians which can write the script and knows all programming language, but don't have the idea of what actually we are looking for in terms of biology, or how we are going to what we need from our data we generate in our lab. So it's beneficial for both of those type of researchers. And so moving on, what we are going to discuss in the next 2 days is transcriptional dynamics, where we are going to understand that what includes gene expressions and what are alternative splicing. Because when you talk about the RNA-seq data, you always think about the differential gene expression analysis, but it's not the only thing which control the gene expression, which is you can predict from the RNA-seq data. There are plenty of things, plenty of questions you can answer from your RNA-seq data. For example, alternative splicing, which is a very common phenomena occurs during the transcriptions. Then I'm going to introduce a little bit about the alternative splicing, why it is important. And then I will introduce you to the RNA-seq techniques and step by step how we are going to analyze the data generated from the RNA-seq, which includes quality control steps, where we whatever you have generate the RNA-seq data, whatever data you generate, you have to pass through that data. You have to pass through some tools such as FastQC to see how your data is, and then you trimmed it or quality control it using the trimming tools. For example, we are going to use Trimometric, and I will tell you as well why we are using the Trimometric. And then we will perform transcript quantification using Salman. Salman is a quantification tool, and as well as mapping tools. So you can do simultaneously quantification as well as mapping with the help of Salman quantification. And then in the next session, we are going to discuss the differential gene expression and the differential alternative splicing using the R-based tool, which is 3D RNA-seq. We are going to use that tool. That tool is available online as well. You can use online as well, but I'm going to use the R Studio, R language, so that we can have more command over our scripts and over our data with the command lines. And then we will discuss about the important mechanism, isoform switching, which is another important mechanism, and then how to interpret the result and how to discuss our result. We can also discuss about that. So gene expression, so it's a little introductions for who are core bioinformaticians, that gene expressions occurs. It's a phenomena from where gene expressed in the nucleus, and from DNA they make RNA, and from RNA DNA transcribed to RNA, and then RNA translate into the protein. And there is a special type of RNA, which is mRNA, which has poly A tail, which translate into the protein. Not all RNAs translate into the protein. There are non-coding RNA as well in the cell, but we are only concentrating on the mRNA because that's the one which translates into the protein. And during the transcription, there is a splicing phenomena occurs where all the exons combine to form mRNA, and that mRNA codes for the protein. And after this mRNA formation, it export to the cytoplasm where they bind to the ribosomal unit and then translate into the protein. So that gene expression regulation depends on number of factors such as transcription factor as well as the chromatin structure. And the gene expression is also specific to the tissue conditions as well as cells. And any change between the two conditions, two cells, difference in the gene expression is called differential expression, which we are looking, differential gene expression, which we are looking from our RNA-seq data mostly. So next was alternative splicing. So you can read one of this review, alternative splicing and protein diversity, which published in Frontiers Plant Science. And more about, if you want to know more about alternative splicings, basically alternative splicings occurs with different events and where you can one of the exons might escape and two alternative exons join together to form the gene to translate into protein. And these are different events which can be occurred during the alternative splicing. So there are number of events, number of types of alternative splicing can occur, and we can predict from the RNA-seq data that which event and which event is occurring in our conditions, differential occurrence of the events as well. That's called isoform switching as well. So if we move on, so what's RNA-seq data? I'm going to give you a brief introduction. What actually is RNA-seq data is? RNA-seq data is the snapshot of the mRNA present in a cell under a specific conditions. So I'm taking the example here, the plants, because I work so mainly on plants. And this can be for animals in a higher eukaryotes, prokaryotes, whatever you are working with. So what I'm going to do is that I will tell you that how we perform the RNA-seq experiment. So you have the plant. What you do is you extract the RNA, total RNA, and during the library preparations, what you need is you need mRNA only because you are mainly concentrating on the gene expression analysis. So you need only poly-A tail mRNA. So with the help of magnetic oligodets, you will extract the poly-A tail mRNA and then you fragment it, perform the cDNA synthesis, and then second stage of cDNA synthesis. After the cDNA synthesis, once you get the double-stranded cDNA, you will perform the adapter ligation, which is depend on which platform you are using for sequencing. For example, the most commonly used sequencing platform is nowadays is Illumina. And with the Illumina, you use Illumina adapters, which attach at the end of this mRNA. And then that adapter ligation, then we perform the PCR and enrichment analysis. And then after the PCR enrichment, you perform the size selection, which is going into the sequence, the size of your library, which finally goes onto your platform where you perform the sequencing.
Sir, what do you mean by when you say library? Can you just elucidate on that a bit?
Sorry?
Like when you say it's preparing a library. So what is it preparing? Like the set of data?
Okay, no. Library preparation is it's the wet work. So when you prepare library means you collect your mRNA, and then before it's going on to the sequencing platform for sequencing, before that you have to use number of steps. You have to prepare a library which load on the sequencing platform. sequencing machine. So that need for that, so the whole process is the library preparation process, and this is the final library looks like, where you have mRNA sequence and the adapters with it. So
Got it. Got it.
the principle is to that's how your platform, sequencing platform, is going to read it. You know, because all mRNA are different. So how they are going to read it, and when you get the data, how would you know that which sequence is of your interest? So adapter is the primer sequence, just a primer sequence, which is need to index your library, and it is recognized by the sequencing platform when they sequence it. Okay, so yeah, actually, I will answer you all these after this session, because let me continue this, because it's going to be long session otherwise. Yeah, so I will give you all the answers after this session once I finish it. Okay. So next step is, once you get the RNA sequencing done, the main step is RNA sequencing data analysis, which is the main aim of this session. Main aim of this session is that how to analyze the data which came from your sequencing. And so there are a number of steps involved in that. And after you use number of tools, and you can perform lots of analysis, and then that analysis finally you convert into the biological meaning. So what happened is, once you load your sequencing library on the sequencer, it will give you a signal on computer screen from the sequencer, and immediately you perform the base calling, which you don't need to do anything in that. Base calling is the quality check of each basis sequence in the machine, on the machine. And it will convert those, with the help of base calling, it can convert those signal into the FASTQ format, which is the raw format which comes after the sequencing. You will get your results from the sequencing platform or sequencing machine. That FASTQ format data is mainly in raw form. So as I said, on the library preparation, you add adapters and primers. So our main aim is, after that, once you receive the raw data, is you trim those adapters. You remove those adapters and just take only that sequence, that mRNA sequence of your use. And then you convert raw data into high quality data, which we are going to perform using the Trimometric. Okay. And before and after this quality control, you perform a FastQC, where FastQC is a program which help you to tell you about the quality of your reads, how good or bad quality of your reads are, how confident your reads are. And once you get the high quality FastQ reads, depending upon what you are looking in your RNA-seq data, you can use different platforms, different tools, bioinformatics tools. For example, if you are to perform the alignment or mapping with your reference genome, you can use Star Aligner, which is one of the tools. And there are a number of tools available, BOTI, HIS2. There are a number of tools used for the mapping. And then, for example, after that mapping, then you perform the quantification of reads. Quantification of reads are that it will convert your reads into the quantification. It will count the number of reads, same reads, and then it will give you a quantification file from which you are going to perform your differential gene expression analysis, which you can perform with the tools like DESeq or AJAR nowadays used. But this is the basic flow chart for the RNA-seq data analysis, and you can use different tools for that. It's just just overview of that. What we are going to use is, we are going to use different tools, which I haven't mentioned here, but yeah, it's depend on your requirement, and I will tell you why I'm using those tools for the analysis of RNA-seq data. So this is the example from one of our paper. This is the example. If you don't have a reference genome, what you need to do for the RNA-seq data. If you don't have reference, for example, Arabidopsis in plants is well annotated, but there are few species, there are a number of species which you are working on, but you don't have their reference genome. So in that case, how you are going to analyze data? And for that, you need to do de novo assembly first, and then you perform the later on the analysis, which is I'm not going to discuss about that. I'm only discuss about the organisms which have particularly I'm discussing about the Arabidopsis which have well annotated reference genome and differential expression analysis only. So how the FASTQ files looks like? The FASTQ file looks like if big text file where have number of sequences on that file like this, these two types, and in that there are each sequence, each read, sequencing read have 4 lanes, 1, 2, 3, 4. The first lane is the identifier which tells the sequence. It is always different from the first another read, sequencing read. So it's the identifier which you will which the tools use to identify it, that which sequence it is. Then you will see a sequence, and in that sequence, then there is a reads as well. It's a forward read or reverse read. So if it is a forward read, there is a plus sign. If it is a reverse read, which is not example, but there will be a negative sign if it is a antisense or reverse read. And then there is a quality score for each nucleotide. There is a quality score provided in the last lane, which tell how good your reads, how confident is that base pair is there at that position. So it's the quality score. So when you open your FASTQ files, it's a very big file containing large number of millions of reads, and of that millions of reads, you will see it looks like each reads looks like a 4-lane reads like this. So this is how you will get. So our analysis start from this point when you get the FASTQ files from the sequence. So first of all, we are going to perform the quality evaluation of that RNA-seq data, especially the FASTQ files, which comes from the sequencing files. And for that, we are going to use FastQC, and I will show you how we are going to use FastQC tool. So FastQC generally is a tool which use for the quality assessment for your data, reads, and whatever reads you will get from the RNA sequencing. And it will give you a report for each file, for each sample, that how is your data is good quality or not, what you need to do in your quality control step. And so everything will tell by the FastQC program. So once you get the RNA-seq data in raw format, the first step is to run the FastQC and then check your data quality and how it looks like. So this is just a snapshot of a flow chart where you can see FastQC files, FastQ files when you perform the FastQC on that. So it will give you all these basic statistics. These parameters tell you per base quality, per sequence score, and all these parameters you will get to know using your FastQC tool. And I will discuss. Yeah.
Sorry
Hello.
to disturb you. How this graph has been generated? It is generated from the Illumina itself, and some other software has been used.
It is generated by FastQC. I will tell you, I'm coming there. I will tell you which tool is used to generate this graph and how you are going to generate it. I will tell you in a minute. Yeah?
Okay, thank you. Okay, thank you.
Yeah. So these are the possible you can get from the FastQC file parameters about your scores. Then we are going to use quality control, because once you get a raw data and FastQC files, you will get to know that how good your data looks. Now, what you need is you trim the adapters which you use during the library preparation in your RNA-seq data, and you trim that using this. There are the number of tools used for trimming, adapter trimming, and quality control. We are going to use Trimometric, and these are the various parameters you can use in Trimometric. We are going to use only the Faraday score 33, which is a quality score that any read be high, less than this quality score, we will degrade, that will be degraded, or that will be removed from your file. And we might use the upper level as well for this score. And these are the just quality parameters. And then, so the good thing about Trimometric, using Trimometric, is it detects the Illumina parameters itself, and it will remove automatically from your reads the adapter without providing the adapter sequence. So it will find out the adapters wherever is adapter sequences, and it will remove itself. So that's why I'm going to use the Trimometric. You don't need to provide any sequence for that. In that, these are the basic command lines used for the Trimometric. And so what you do is it's a Java-based command line, and then you just perform the Trimometric. You give the command line. You can perform, depending upon your data, it's a pair end or it's a single end. If it is single end, you use this one. If it is a pair end, you use this command line. And we will provide you this command line as well, scripts as well for your sample. And then you put your forward and reverse. If it is a pair end reads, you put forward and reverse reads. You give the path of the output files. And in the output, for each read, it will generate two files. One is paired and one is unpaired. So what's
porque el que tiene este bull es esto. Parado en la pantalla.
I request everyone, please mute yourself, because hello, please mute yourself because there is a very too much background noise coming. I request everyone, please mute yourself doing this. So yeah, so you have paired end and unpaired. You will get two output files as a paired, unpaired for each read. And what does it mean? In the pair end reads, the paired means is only those reads whose reads are found in the forward as well as in reverse pair. Whereas if there is some reads which the tool don't find the pair, so it will compile in a unpaired reads. If you don't have sufficient read in your pair end files after the quality control, you can use these unpaired reads as a single end reads in your analysis. And usually we don't need those unpaired reads. We need only the paired reads. They are sufficient for the analysis. What Trimometric uses, Trimometric use the uncompressed or gzip file either of the pair end and single end reads, what is called raw reads you get, and it will give you the output file as a pair end or unpaired reads. And for single end, it will give you only one output file for one read, individual read. Then we are going to perform transcript quantification using Salman. And why we are using Salman? Salman is a very robust tool used for the quantification nowadays in number of RNA-seq analysis. And why we use Salman is it's ultra fast. It requires a very low memory. You can run on your normal laptop, and it can quantify and map your expression of your genes simultaneously. You don't need to first you generate the mapping files and then quantify them. It will do simultaneously both the things, and it will quantify why it is ultra fast and low memory required, because it requires the transcripts only. It doesn't require the whole genome. It requires the transcriptome data only. With the help of that, it will quantify the expression of each transcript. And there is a mode called quasi-mapping where it will quantify the expression of your gene or transcripts simultaneously as well as mapping. And it will give you a mapped file in BAM format, as well as it will provide you the quantification file. So we are going to perform the differential gene expression analysis and differential alternative splicing. For that, we only need the quantification files. We don't need the mapping files. And what the requirement is the index transcriptome reference genome, and FASTQ files, high quality FASTQ files for the quantifications. So these are the different parameters you can use in Salman. One is index, which require for the indexing of the reference genome or transcriptome. And the other one is the quant, which requires for the quantifications. And there is a new features comes Alvin for single cell analysis. And there are other features as well, swim and quant merge. We might don't need those ones. Quant merge you can use when you combine your replicate file, if you want to combine your replicate files, bioreps or technical reps. So
just a question before you start, sorry.
Yeah.
We have a lot of data in TCGA, you know, that RNA sequencing data, right?
Yeah.
So can we do the same on that data, or we can't because that is produced by them?
TCGA data you have, but
yeah,
you can perform the Salman quantification in that, but you need transcriptome data.
So you mean the BAM files?
No. The reference you take for the TCGA. So the reference you take, genome reference you take for that. In Salman quantification, you need transcriptome data, not the genome data, not the DNA files. You need a FASTA file for the transcriptome. Okay, that's the only thing. It will give you the expression of the genes or mRNA, because it takes only the transcriptome, not the long gene sequence as a reference.
I'll need to get the FASTA file for that.
Yeah, FASTA file.
Okay, thank you.
Yeah. Okay, before we are going to move forward, let me close my pointer. Oh, skip this. I'm going to stop my slide presentation, and I'm going to start with the analysis part now. Okay, bear with me for a minute. So let me open the files. Scripts. Okay, so you have your system now. On your system, you can open the window, terminal window, where if you are working on Linux system, and yeah, you require the either Unix command line system for this analysis that I forgot to tell you. And if you don't have a Unix system and Linux system, and you still want to perform on your Windows laptop, so what you need is you need to go through the Galaxy. And with the Galaxy user, you can generate the quantification file, Salman quantification files. And those Salman quantification files we are going to use in the next session in the R. You transfer those quantification files in the R session, and where we perform the differential expression analysis. So Galaxy is straightforward. You just upload your data there, and Galaxy you might use before as well. And then it has Trimometric, it has Salman, it has FastQC, all the tools available on Galaxy. It just needs some time when you upload your data, and then it will perform the analysis. And then with that analysis, the output file generated in that after the Salman quantification, you can transfer that into your R studio, which we are going to discuss. Yeah.
Yeah. What is Galaxy?
Oh, Galaxy is a online server tool for the. It's a platform. You can use it if you have good internet connection. Let me just.
And the way you're doing it on your this terminal, can we do it on Ubuntu?
Yeah,
Okay.
you can do it Ubuntu, but if you have, for example, if you have Windows, then what you can do is, if you have Windows, then you can't run the Salman on Windows. So you need the
So Dr. Saurabh, the quality of assembly and other Trimometric and like other things using by the Galaxy, and when we are using on our own system, giving the same type of result, or there might be a difference between the Galaxy and the.
Yeah, actually, if you are performing using your own command lines, you will have more independence of selecting the parameters. With the Galaxy, there are parameters which are fixed, which you can't change it. So, but for this kind of analysis, it will work.
I have used the Galaxy many times. Using the Galaxy, I have done the assembly using the Trinity and Trimometric and like other things. But sometimes many people say that the result generated by the Galaxy is not giving the as much as transcript which are using on the Linux and using the Trinity and other thing.
Yeah, I agree with that, because you can't do much with the Galaxy. There is, but if you have data and you perform the fast QC as well, if you know that your data is good quality, even in the Galaxy, so there is no need to worry about much that you are doing with Galaxy. And if you don't have if you don't want to use Galaxy and you want to use the command line Unix system, and you have laptop only, which is Windows running, you can ask Decode Life. I think Parveen is with us, and you can ask Decode Life. They have a server. They will provide you the access for that server as well. And you can use their server for your analysis, your data.
Okay.
You can use that.
Who will provide this facility? Who is providing this facility?
You just email to the Decode Life.
Sorry to interrupt you, Dr. Saurabh.
Yeah, sir.
Hemant this side. So due to some maintenance issue, that server was down and it's not available in this workshop.
Okay, it's fine. We are not talking about this workshop. We are talking about in future, if somebody.
Yeah, in future, definitely.
Yeah, I'm just saying in future, if you want to use your Unix system.
Is it free?
No, it's not free, but it's a minimal cost it can provide, because server can't be free, because if you have to pay lots of money to develop a server. Yeah.
Okay, thank you.
So I don't know much about that, because but you can get in contact with the Decode.
Okay, I will get in contact. Okay.
Yeah. So you use the Unix command line, or you can generate on Galaxy. I just send a link on Galaxy link on the chat box. You can use that for your generation of quantification file. So open the terminal, and we use one by one the command lines, and let's go use that one. Okay, so first of all, in this analysis, what we are from where we are going to use, what the data we are going to use is. So I'm just going to show you this text file as well. So you can see the text file. So this is the paper from where we are going to use the data, and the data is available on SRA, NCBI SRA under the study number this. So you can download the data if you want to work on this.
Excuse me, can you just maximize the screen, please, because we're not able to read. It's too small. Thank you.
And could you please paste this link, this accession number, in the chat box so that we can download the data?
Oh yeah. So under this study, there is a number of data available, but I'm going to use only the Arabidopsis Columbia lines data ecotype, which is grown at 22, and we provide the 4-degree as well treatment. So there are two conditions. One is control, which is 22 degree, and there is a data of there is a stress samples as well, 4 degree. So we generated the RNA-seq data for this Columbia line, which is a wild type of Arabidopsis, under the condition of 22 degrees Celsius and 4 degrees Celsius. And why I'm going to tell you, why I'm telling you this is about this, we are going to use in the next session, not in this session, but we are going to use in the next session, where we are going to use the differential gene expression analysis between two conditions. That is, one is normal control and one is cold stress, 4 degrees Celsius. And for that, each sample, we have 3 biological replicates, and I will tell you why we are using the replicates, what is the significance of using the biological replicates. In addition to that,
I have a question. Like from SRA, we will get the binary files or directly the this FASTA files?
You will get the binary files, and then you have if you have a SRA tool, you have you can use the there is a dump command, fastq dump, which converts those binary files into the FASTA files, fastq files.
Okay, thank you.
Yeah.
One more question, Dr. Saurabh.
Yeah.
Is it suggestible to run these the whole set of the tools over a laptop? You need a server to do this analysis.
For this, it depends on how big your genome is. For genome size like 1.35 MB for like Arabidopsis, I use the normal laptop.
Okay.
But if you have a human genome or you're working on big genomes, then you need a definitely big server. But I think you can run it easily on your laptop. These command lines. That's the good thing about Salman is Salman quantification. That it takes very low memory of your system, and it's quite fast. The only requirement Unix. You need to have Unix command lines or Galaxy. That's the only two things you can have options. Okay.
Yeah, thank you.
So right now there are 44 data sets.
44 data sets for?
So like I put this as accession.
That's, I told you, just we are not going to use all. We are going to use only the Columbia. It has a methylome data. It has a nucleosome data in this paper.
Okay, thank you.
So we are going to use only the RNA-seq data of Col0. And you don't need that, actually. I'm just telling you the difference between the data, that it's 22 degree and 4 degree. I'm using that data. But it depends on your data as well, what you are using, what you are working on. Okay.
Sir, excuse me, sir. Sorry, sir. Under this SRA ID, we have 44 results. So which one you want us to select and download?
I just told you.
So I'm so sorry, sir. Actually, we're looking on the.
Yeah, I just told you. This
I guess
only
different system, the results are loading in a different pattern. So that's why it's hard to find.
So it's just Columbia lines I'm using from that data. There is only these line RNA-seq data of Columbia line grown at 22 and 4 degree. Okay. And each line has these 3 biological replicates and technical replicates. So
Sir,
for
any other heading? Because we are getting like MNA sequence of April.
yeah, if you keep searching, you will find RNA-seq as well.
Okay.
Yeah. So there are a number of data on this. So that's the study equation.
Grown at so grown RNA-seq of grown at cold temperature.
Yeah, cold as well as normal.
Okay.
So we need to check each and every data because in this page I think there are multiple data. To which data should I select?
Okay. Okay. Before you're going to search that data, I'm not saying that you start working on that data.
Okay.
Just let me tell you. I'm just saying that data I am using it for your information in this session. Okay.
No, I mean to say that how I will because the SSN ID you have provided in chat box. I think here multiple data are designated same ID.
I think, sir, if you can show us an example of like go to the SRA and kind of just download. If you can show
actually,
small example, please.
It's this data is generated by in our lab and it's available on SRA, and it has a number of files like RNAseq, bisulfite sequencing, RNA sequencing. So I'm using only the RNAseq grown at 22 degrees Celsius and 4 degrees Celsius, only the col0 ecotype. So if you are facing any problem, let me show you then.
Just you can show how we can download so that we can also
I can't tell you how. I can't tell you because it needs a command line. It needs a SRA tool to upload it here. Okay. So I can't tell you here because it takes time to download the data.
Okay, sir.
Yeah. Sorry for that because it's not it's not included in this session, to be honest. It's just because the main aim of this is your data. It's not about the what are publicly available. It's what you are working on. Yeah. So it's not like that there is a loss of data available and you can use that data. You can download that data and work on that if you are is of your interest. So, but yeah, for downloading, you use a SRA tool, Unix command line, SRA tool. You need it to download any data from NCBI. You need SRA tool, and then you download the data and convert that into the FASTQ files. Okay.
Okay, sir.
So it takes like a file needs like almost, if you are downloading on your laptop, it takes like 2 days to download one file. So it's not like I can't cover this in this session. So you can see this RNA-seq col0.
Yes.
Grown at cold temperature. Makes sense. Yes. So it's a 4 degrees Celsius. So all these are different replicates of cold, and the normal temperature is 22 degrees Celsius. So we just need col0, not the apparel. Just col0 I'm using, normal and control. That's it. And this data is raw data. So this data is raw data. You have to perform your analysis, all quality check and everything. All data available on NCBI SRA, you have to go through the quality control. Okay.
Okay, sir. Thank you, sir.
Okay, you're welcome. And yeah, so just keep going that. So it has replicates, blah blah blah. Let's talking about the. So the first step is you have FASTQ files. Let me show you the FASTQ files as well. Okay. So here I'm using only one replicate, rep1, and these are the forward read and these are the reverse read. This one is reverse read. So this is only one sample, one rep, one read, pair end. So this is read, forward read. This is reverse read. I'm going to use this one in this session. So it is present here. So what I'm going to do is I have FastQC downloaded in my system. It's a Java-based script. You just type on your terminal FastQC and put enter. When you enter the FastQC, there is another window opens like this. So this is FastQC window is open. Now you can open the file wherever your files, these FastQC files are. So my files are on desktop in a folder RNASeq, raw reads. These are my files. I'm going to select both files and open them. So you can see the files are loaded here already, and depending upon the internet speed, it has started analyzing it. Okay, so I think I don't have very good speed here today, but yeah, it will start analysis. These are big files.
So, Dr. Saurabh, it is not working offline.
Sorry?
It's not
It
working
works offline. It works offline. FastQC file works on FastQC works offline, but the report, if you want to download it, once you get the report will be you need to see the reports. You have to save it. Otherwise, you need the HTML server to read those files.
Okay.
It also depends because my system is bit slow, so it doesn't have much memory, and my data is really big. So,
sir, if we want to download any other data, suppose, and analyze the FastQC thing in Linux, so like I will have to get this FastQC thing also installed in the like through Ubuntu.
Yeah. FastQC Ubuntu. You can do FastQC on Windows as well.
Okay. So is there any, like, can you tell me the command to get it?
Well, I use always Anaconda command line. So for Anaconda, it's easy because you don't need to provide the path every time for your files. It's always easy. And if you just type on Google Conda install whatever tools you want, it will give you the command line. You just copy paste that. Let me show you. So Conda install FastQC. So you open this. And, so yeah, you can use this command line. Just copy paste in your terminal and yeah. So whatever tools all tools I'm going to use is using Conda. And I think you have the Linux session as well in this training program. So you must be aware of the command lines as well. So you can download it from their respective website. If you see, I will provide you this. These are the respective websites of the tools using this. Okay. So you can use this. So it's 25% done. Meanwhile, if you have any question, you can ask me because I can't read
these sequences, as you told that they are, in a SRA in NCBI. So we need to download SRA toolkit to have these converted into FastQ. Then we need to also have a FastQC software to run.
Yes. SRA toolkit is just to download the data from the NCBI.
And this TCGA data which we have, they have level 1, level 2, level 3, level 4 kind of.
Yeah, I'm not sure about the TCGA data, how to download it from there. But because I don't work on cancer. Yes.
In your case, the data has been generated in your own lab, right? But if for our case, we want to select some data for that, is there any particular pipeline or data selection process? Because as earlier also it was told that there are so many data available, and then there are so many variants of the data available. So how can we precisely select data? That's the first step is very important, according to our research.
No, you will get the result after you analyze the data. So what you are looking for, you should select the data according to your requirement, according to your query. It's not depend on that you will select any variant or any data. There are a number of data. What you are exactly looking for means you read some paper and then you find the data there. They always give you the SRA number. So that data you want to work, you can download that data. What does it mean according to your result? It's depend on your interest. You know what you want to do. Yeah, somebody is raising hand. I can't see it. Can yeah. The one I'm using here. The one I'm using here is the terminal. Fastest. I don't think it makes much difference fast QC, but your terminal is faster than Galaxy, because in Galaxy you have to upload your data first, and then after when you upload your data, then you just give the command line and you just go offline. It will run there on the Galaxy, and it will, once it will finish, it will give you a email. You have to put your email address that your data is completed analysis.
Actually, on the Galaxy, at a time there is a lot of queries and lot of the job subscription is going on.
Yeah,
that's why they are taking too much time.
Okay, yeah, that might be. Yeah, Shalini is raising hand. Yes, Shalini.
Sir, can you please tell how we can perform in the Galaxy? Because as I am having a Windows and using WSL Ubuntu, I'm not getting the correct output.
How to use the Galaxy?
No, just, I know how to use the Galaxy. Just a simple like how we can download the SRA file and all this.
I don't know about that. I told earlier as well. I don't know about how to download the SRA file on Galaxy. So I will suggest you download the SRA file on your system. Even if you have Windows, using the SRA tool, you can download on your system and then upload from your system to the Galaxy and then perform the analysis.
Okay, sir. Yeah. And sir, I use the Ubuntu in the using WSL.
Mm-hmm.
Can I use it?
Yeah, you can use Ubuntu.
Okay, and the command is the same for the Linux and
yeah. I'm using Mac, so but the Unix commands are same in both.
Okay, sir. Thank you.
Yeah, except Windows. Windows commands are different always.
Yeah.
Okay, Gaurav. Yes, please.
Yes, sir. Good evening, sir. Sir, actually, I am currently pursuing masters and I have RNASeq data to do research. My first question is, sir, you said about Trimometric, but we are trying to do by FastP. So can you differentiate between two? And you have mentioned in the pipeline about star also. So we are generally interested to do at HiSat 2. And please differentiate between star and HiSat 2.
Okay. All right. So if we talk about the quality control, you can use any tool, whatever tools you are comfortable with. As long as your data is good for perform the analysis is fine. So there is no such thing that which one is better for the quality control, Trimometric, TrimGalore or FastP, whatever you are using, MultiQC. So whatever, there are a number of tools available, so which you have easily access and which you are comfortable to use as a command. So you can use anyone. So there is not much different. The main objective of that is to remove the adapters, to convert your raw data into the high quality data. That's the main aim of your data analysis. Okay. So
okay, thank you, sir. About star and HiSet too, can you please elaborate?
Okay.
Sir?
Yeah, star. I'm not going to use the star. Why I'm going to use the Salman, I will tell you in the next session why I'm using that. But star is a soft tool which you can use for any samples, any organism RNA-seq data, but it's mainly used for if you have a big genome. So star has a big so you can use a star aligner. It will give you the almost same result. But in the star aligner, the only thing is you have to perform first the mapping. There is a two-step procedure. You have to perform the mapping, and then you have to perform the quantification. With the Salman I'm using is fast, and it will perform mapping and quantification simultaneously. But if you have a big genome, reference genome, I will suggest use tools like star or HiSet2. In HiSet2, you have it's a advanced version of Botai mapper, HiSet. It will give you more details of splicing as well. In Botai, you can't get that splicing details. So HiSet2 is equally good as star. So it depends on you which tool is comfortable.
Thank you, sir. So when you say it is a big data, means how much file you are, sir, referring to? Sir?
Anything bigger than Arabidopsis. Arabidopsis is 1.35. So I think bigger than that. For example, wheat. You can use star if you are working on wheat. You can use.
I am actually animal.
Yeah, animal. Of course, animals have comparatively big data as well. Humans, especially if you are working on humans, rats. So yeah, star aligner is good if you're working on that. Okay, we got the files here for this fast QC. So I gave the. Oh, I'm sorry, just my system is. Anyway, let me close this. So I gave the raw files to this fast QC, and it gives me the basic strategy. The good thing is that my raw files looks really good. So if you see here, these are the basic things. So these are the number of sequencings it analyzed, and it will gives you the sequencing length, percentage of GC, all these parameters you need it to know. Then this is the basic studies. Then if you click on the parameter, you can see in my data. So now somebody asked when I was presenting these slides that what are these boxes? So here is the boxes. The reads which line in the green, that's the quality score. So if you see the quality score is 28 and more, is in green line. So if your reads fall, all reads falls on the green line, in the green part, your data quality is very good. If it comes to the orange, like some of these here, reads, these are the positions. So some of your base pairs are of quality less than 28, and it comes in. You can use that data. It's not like it's a bad quality data. It's average quality data. But if it is in green line, it's really good data. If it is in orange line, it's a average data. You can use it. But if your data falls in exactly in the red line, that will not at all accepted. So any data, it's not, my one is very pretty clear actually. It's not coming anything here. So we can't use that. Then you have to perform further quality checks like Trimometric, I'm saying, and then you have to trim your data accordingly. So this is the purpose of this graph per quality. these are just the same thing representing in a tiles format. If you want to read it, it's no sense, but it's orange, you can accept it. If it is red, then yeah, you have to perform the quality scores, quality checks. Per sequence quality is very good. Like per sequence is, if you see it's increasing, it's going mean quality Farad score, and it's the most of the sequences are 38 Farad score, which is good quality score, and it's green. If it is red, then not good. Per sequence content is quite different because these are different, like ATGC with different color. So it's not good here, means your GC content is either less than 40% or something. So that's, yeah, it might be the problem of adapter because you are attaching the adapter. So after removing adapters and trimming, it will get fine. It will, your sequence get fine, will be fine to analysis. But per sequence GC content is perfect here, perfect green, like nearly 50% if you see, is perfectly fine. Per base and content is fine. Per base and content means the content which are the base pair which are not confidence, that sequencer didn't recognize that is 8, T, G, or C. So it's N, it's perfectly fine. There is no N there. And land distribution is also good. Sequence duplicate lesser, there are duplication of the sequences, which is fine. We know because it's a raw data and you have adapter contaminations. So there might be those sequences which are repeating again and again in your file. Over representing sequences, there is no any sequence which are over representing. And adapter contamination, there is no adapter contamination. It looks like that my file is quite fine, pretty control. So this is how you are going to interpret. So this is overview of your RNA-seq raw data files or high quality reads data files. So this is nothing but just for your confidence level that, oh, my data is good. I can perform the downstream analysis from now. Okay. So there is no must to that. It will tell you about your quality. So both the files. The second read is still going on, so I'm not going to stuck with that. It will give you the same file. So this is how FastQC works. And if you close that FastQC files, so now what is next step is, once you have a contamination for that adapter or your raw sequence is not good quality, what you're going to do, you are going to perform the Trimometric, which you can download it from here as well as from Bioconda. And this is the command line, which I will give you. So you just need whatever sample you are using, you just need those sample names. You just replace the name of this sample with your sample and you perform the analysis. Just if I copy, paste Trimometric here, run it, it will start. It will take time, nearly an hour for one file. So I'm not gonna wait for that. After this, you will get 4 files. Let me tell you the 4 files for one. I will show you what file you will get.
Sir, sorry to interrupt. We
to
download Trimometric software as well. To
all the tools software you are using, you have to download all.
Okay, and Trimometric will also run on Linux itself.
Linux, yeah, they are requirement. FastQC can run on Windows, but yeah, requirement for Trimometric is Linux, Unix system, or you can use Galaxy as well. There is a Trimometric on Galaxy as well.
Sir, so the purpose of Trimometric will be doing a quality check, or like for the mapped reads, pair reads, as well as.
No, it's not about the mapping reads. It's just a quality check. So your raw reads, which you come from the sequencing or you download from the SRA, they are not a good quality reads. They have adapter contamination, they have sequence replications, and they have lots of things. They are not short reads, they are too long reads, too short reads. There are a number of things. So you have to trim them, you have to polish those reads. The function of Trimometric is to clean your data, raw
Okay,
data.
sir.
Yeah. So I will show you how it looks like. So you will get these 4 files for the paired and unpaired for each read. So these are paired reads and these are unpaired, which are present in your sequence but not paired with each other. So for the forward and the reverse, they have paired, but there are few reads which are unpaired. So what Trimometric does is it can take all unpaired reads and make another file. So if you have sufficient number of sequences, for example, and you will, and if you want to require more reads, you can use as these reads as single end. But yeah, pair end is sufficient number of reads you will get in your data, and then you can perform analysis with these pair end reads. So this is the file.
Okay, so these files, these FastQ files you have downloaded from the Illumina software, the Illumina data. And then basically you are trimming them, the unpaired ones, you're trimming them, you're running them on FastQC file to see basically. So like it's like how is the chronological order decided? First you download FastQ files from the Illumina software. Then you run
Illumina is not a software. Illumina is a sequencer machine.
Yeah, the software whatever Illumina is using, I'm not aware of that. In Thermo we use torrent suite software, torrent. So in Illumina you must be using any software which is compatible with Illumina. So the first one is you are downloading these FastQ files from the software.
It will come to you as a FastQ file from your Illumina sequencing, torrent, whatever sequencing platform you use.
Yes, sir.
You get those files. After you get those files, you will check your quality using FastQC.
Okay.
If it looks good, you can go directly to the Salman quantification.
Okay.
Mapping or quantification, whatever tools you are using. Okay.
Okay.
If it is not good quality, then you have to perform Trimometric or any other quality control tools like TrimGalore, or you can use CutAdapt, you can use FastP, you can use MultiQC. So whatever tools you want to use, you can use that tool to trim it, and then you will get the reads which are good quality FastQ files in the same format, FastQ format, and then you use them as from mapping and quantification. Okay.
Done. Thank you so much, sir. Thank you.
So one thing I would like to ask here is that as you have selected, there are multiple replicates, including the control and the stressed tissues. So you have demonstrated here that you have selected the depth one only, or you can say the normal rhabdosis in normal temperature, and you started the analysis part. So how we can compare the stress data with the control sample? So we need to go for we need to go for the analysis each and every replicate. Repeatedly. Suppose we have completed the one part, one replicate, then we'll go for the second replicate. When we will go for third replicate,
fourth replicate, and fourth sample, fifth sample, sixth sample. You have to do it for
in the
individually
same way for stress tissues. Okay.
Yeah, because I can't cover in an hour.
Okay.
So I just take the example, just select one file to show you how it works.
Yes, sir. After that, how we will compare the stress with the control samples?
So like this, you will download all the 22 files, 22 degrees Celsius. You will download all the 4 degrees Celsius file. You will perform for individual files, FastQC, Trimometric, and once you get the high quality reads for each files, those files will go individually into the Salman, and then you have to perform for each file, each sample, each condition. It will generate a quantification file. Okay.
Okay.
So yeah, here I'm just showing you the one example. That's it.
Okay.
So you might have 50 samples. You might have 50 with 3 replicates, means you have 150 files. You have 200 files. You have to perform individually with each file.
So is it possible to collect all the normal samples and
Yes.
upload, or you can process simultaneously, or we need to go for an individual and one sample, one for
yeah. It's a thing. You can give the command line to your system, all samples together.
Okay.
Yeah, you can't go one by one. You can give command line, and if you are using Unix system, I will tell you tomorrow that you can make a executable file. So what the executable file does is you can put all your command lines for individual samples in that executable text file. You save that file, and in the command line, you just run that file. So in that file, once the terminal will take each command individually, step by step, and perform the analysis. So you don't need to go, oh, my one sample is done. Now I need to put another command line. No, you just give the command line, forget it. And then all will done simultaneously. There is no such command line that you can perform only one single command line for all the samples. No, you can't do that.
So I think, would you like to demonstrate the same?
I will tell you tomorrow.
Okay. Thank you so much.
In tomorrow's session, I will tell you that how to generate a executable file. Okay, but I think, the session is already going out of now time. So if you have, sorry, I can't read the chats because this number of messages there. If you quickly have questions, we can take a couple of questions more. And then if you have, I just writing my email address. You can email me if you have any questions, and then I will get back to you. Okay, just if you want to ask till now.
Hello, sir.
Don't worry about script. We will share the script with you.
Yeah, okay. Don't worry about the script. We will give you the script as well as presentation.
Yes, sir. So that was a wonderful lecture from you. But there's one thing I needed to ask.
Yeah.
That the number of steps are too many for me to comprehend. So
okay.
Yeah, so it would be better that we go for a workflow first, and then we do the analysis using that workflow one at a time.
Okay, can you see this file, text file?
Yes, definitely, I can see that.
Okay, I will give you this file.
Yes,
it will give you stepwise. You have to perform quality check with FastQC. From where do you download FastQC? Then Trimometric. And then after Trimometric, you have to perform Salman quantification.
Quantification. Okay, and then we have the data. Yes.
Yeah.
Yes, thank
so
you so much.
At the end of this, whatever the how long is going to be, I think the decode will provide you all the scripts and everything step by step. Okay.
Okay.
Sir, Salman can be used for aligning also with the reference genome.
Sorry.
Salman can be used for aligning also with the reference genome.
Yeah, if you do the mapping, validate mapping in the command line as well, it will also give you the mapping files, BAM files as well.
And variant calling, sir.
No variant calling.
No variant calling.
No, variant calling is a different thing. It's completely different thing. You need different thing. It's just to give you the quantification file, which I am going to use it tomorrow in the R studio. I will move those files. I will just show you those quantification file, how they look like, how to transfer into the R, and in R, how to perform the differential gene expression analysis and other stuff with that pipeline, and how to interpret your results. Okay, so if you want to do these analysis, you have some RNA-seq data your own, or you can download from there and just try it. If you have any problem, you can ask me at any point. Okay.
Excuse me, Dr. Saurabh, can you suggest some of the tools for de novo assembly?
De novo assembly, there are a number of tools available for that. Abyss, Trinity. Trinity is good. Abyss, SOAP de novo.
Spades is another one.
Yeah, I used around. Okay, that's okay. Quickly, I'm give you one of our another paper reference where we use around 6 in the chat box. We use 6 tools, de novo assemblers, and you can use any one of them according to your requirement. So I just shared it on the chat. You can go through this paper and this will give you, we optimize the de novo assembly.
Thank you, Dr. Sir.
Yeah. Yeah, we commonly use this Trinity and SOAP de novo. Yeah. So I think it's going to take time, and I already told you how Trimometric works. So tomorrow from where I'm going to, I will not perform this command lines as well for Salman because it also takes little time. So these are the command lines which you will get. So I will quickly tell you what you need for that is. So I'm using the Arabidopsis transcriptome data, RNA-seq data. For Arabidopsis RNA-seq data, I'm going to download the reference first. To download the reference, there is a ATRD2 reference. Why I'm using this reference, not TAIR2, is that because it has more number of alternative splicing transcripts than the TAIR10. Okay, so I go to this website. There is a FASTA file here, .fa, and one GTA file. I download these 2 files. Just click on this and it will start downloading. And that's our reference files for our quantification. Then I will index the file using this command, the reference genome, where you give the command. I will show you how to index the reference genome tomorrow, and then you start quantification using Salman. I won't run this tomorrow because it takes time. Salman quantification, it might take a couple of hours for analysis. So yeah, and the thing is, I will tell you about the executable files, and then we move to the main part of that differential gene expression analysis. Okay, indexing in easy language. Okay, I'm giving this because it's getting late. This is the last. If you have any other query, you just email me. I'm going to answer this one. Indexing your references. The reference genome is always big. Transcriptome data is always big. So, and there are millions of reads. Okay, so if you every read start going to map and quantify at a one place, it will take years to complete that data. So what these tools are doing is, these tools are too smart. What they are doing is they break that sequence, that genome reference sequence, into small files, small index files. That's called indexing in easy language. So making reference genome in multiple files. So suppose if you have a big file and you are searching a word in that text file, it will take a while. But if you have a small files, you will quickly search those, or where it map, where it align. So that's it. So that indexing is reference genome is to break down that whole genome into smaller files so that it can be fast, quick to perform. Okay. That's okay, Shobita? Yeah, indexing is just small files. Convert the big genome file into small files. That's it. And then we will perform the quantification. Quant is simply quantification, how many reads are present for particular gene in a file, and then we will do the 3D RNA-seq analysis using R tools. Okay, I think I'm going to Andres,